All righty. I believe that we are live. Hey, everybody. Thank you for joining. Today is a very exciting day where we get to announce the winners of the BioXAI Hackathon. Just want to start off by saying that, quite frankly, Sean and I were blown away by the number and quality of submissions that people put forward. Thanks for bearing with us, giving a little bit of extra time to judge. It was, I think, a little bit trickier than what we had originally thought, trying to figure out the right folks to actually highlight both on this stream and to award prizes to. We are very excited for what comes out of this, both from the standpoint of Hackathon results and from the standpoint of what comes out as a larger BioXAI community. So we've got a quick few things that we want to talk about before we officially announce the winners. Sean is going to, I'll share my screen, and Sean is going to give us just a quick overview of bioagents. We're going to look at some of the themes for the submissions that we saw. And then Sean's going to talk a little bit more about the open source community for scientist bioagents and kind of the whole purpose behind the hackathon. We'll go into announcing winners. We'll give a quick chance for some of the sponsors to explain how their tech can be used by people building bioagents and at the intersection of science and AI. And then we'll go into some quick demos for each of the teams that are there. So with that being said, let me go ahead and just quickly share my screen. I'll show these tips again. Easiest, yep, okay. So Sean, I'm going to go ahead and share this. And if you want to quickly talk through, I think that can work.
Perfect. Just been really impressed by the submissions. Like over 60 projects submitted over the 2-month-long virtual hackathon. A lot of different topics and subjects, but I'll really want to, what we're going to share, because I can go into the initiating idea of bioagents and how we're turning agents into both scientific task runners and tools for scientists to use. Cool, yeah, I see it there. How agents are changing, for anyone tuning in that hasn't been watching the agent space in the past, well, I'll start about 8 months ago is when like the Web3 Twitter agent space really took off with Virtuals and Eliza and mini projects having their first releases. Of course, agents have existed for a few years now, depending on the capabilities that you ascribe to an agent. They're still getting better, but they're now reaching the point where they can do long-running tasks and also think at a PhD level. So when you combine the two, they can think for a PhD level for a very long time. And so bioagents were the first attempt to take a turn-based conversation agent, which is what you're used to with ChatGPT and things, where you say one thing, it says another, and you repeat, and returning that into a task runner agent that is just running continuously or for a set amount of time on a specific scientific task that you give to it. And so I think we're one of the first to do this initial change to how the ELIZA framework is set up, where instead of doing turn-based conversation, it's doing these tasks. So bioagents have been around for a few months now. And when this hackathon started 2 months ago, they were very new. And so we appreciate all the teams that helped both in finding bugs and improving the state of the code quality, and working with us and with the ELIZA team as the ELIZA team goes through a giant expansion and growth and some changes to how their agents are set up. So we appreciate all the hackers that worked with us, and maybe we can go to the next slide here. And so I talked about these agents, and two things that are most important to think about is they're is the concept of plugins or packages for agents, where one person can design a plugin similar to working with like an MCP server that lets the agent do a new form of action. And so these actions could be calling other APIs. They could be saving state to a knowledge base. It could be interacting with a knowledge graph. And plugins, I see, are what's missing from some of these agents, where if you look at the Web 2 science space, there's a lot of companies now, Google, Microsoft, Future House, there's a lab from Stanford called Biomni. All these more like Web 2 scientific agents are improving their capabilities through adding new tool usage. And so one of the goals for this hackathon was to create plugins that added to the agent's capabilities through giving it the ability to interact with more types of scientific tooling. And secondarily, and even possibly more importantly, is knowledge graphs, where we think that agents using knowledge graphs are going to be extremely important for how the open source agent science space develops. And so we'll talk about a few ideas we have, and we saw some great teams working with knowledge graphs in the hackathon. Next slide. And so we had really amazing sponsors. Solana, here's on the call, Prime Intellect, Molecule Labs, and Origin Trail were amazing gold sponsors. Thank you all. And some other sponsors we had are, you can go next slide. VitaDAO, CerebrumDAO, Beard, Coordination.network, and Eliza. And so all the sponsors were extremely helpful. Thank you all for being there for the Discord office hours and helping out the hackers. And yeah, really appreciate all the support. We also had some amazing judges and mentors, and they were hosting office hours throughout the 2 months. And we, super can't explain enough how appreciative we are of your time and advice and expertise. And hackers, if you had any good office hours with the mentors, please both say thank you, but I hope the mentors would consider themselves a long-term resource as you continue to build your projects. And moving on, we had around 60 projects built. 11 finalists will receive a prize. We'll go through the prizes coming up in a sec, and we'll have a few demos for you as well. So now these are the demos that we showed off last week at DSci Berlin. We had an event in person in Berlin, and there's these videos here. We can go.
And Sean, yeah, with the 3 that we shared in Berlin, we do have the videos of those queued up, but I think we might even have a couple new ones to share a little bit later in the call, which I'm pretty excited for. Maybe before we go into some of the sponsors and the tech that they are building on and kind of giving them a chance to explain what they're doing, can we maybe just talk a little bit about the open source community and kind of the intentions for the event itself, and some of what we hope to see coming out?
Yeah, we have. So the idea of AI for science is a fairly new one. Well, it's been around for a little while, but it keeps improving and improving. I joined Bio in December to grow a new team around AI at BioProtocol. The website's bio.xyz. Of course, if you're on the stream, you're watching it on X. But the concept of AIs can be assisting researchers, or another idea is more autonomous steps where the AI is taking doing a multi-step process of knowledge work to output a result that would take a person either a lot of training or a lot of hours. And so we're building in the space of agents to improve researchers and accelerate the pace of science. And I like to say, like, increase the throughput of thought, where these agents can be applied to a specific problem that a researcher has come up with. If you've used O3 research or deep research or these others, like, you can actually kind of delve down into an idea and go through the idea maze. And we're creating agents that are helping with that. And so it's really there's so much that it can do. Like, there's so many different scientific toolings, so much to add to it to really make them capable enough to help a postdoc in a lab or someone at a pharma company, anyone starting a biotech company. These types of agents are perfect for that, where they can remove some cognitive load and help them get results faster, get to experiments faster, maybe, and help them with micro proposals. So we've started this here at Bio, but we wanted to make it a community. And of course, ELIZA has an amazing open source community. Origin Trail is open source. And we want to also be part of this open source community and grow it. And of course, we're putting in the paths and the structures so that as the agents grow, as people contribute to them, the contributors are both rewarded and the agents themselves can have a token part of it. But yeah, the open source community is just getting started. We have the Bio GitHub that we're continually adding to. We're continually growing the team at Bio, and we'll be adding in more specific processes in place to both reward and give some accolades to the open source developers that are contributing to the Bio repos. So for anyone who's listening or anyone who has developer friends, we are accelerating what we're doing here at Bio. And so we'll be sharing more links. We have the public GitHub, of course, but if you have any specific questions, I can always talk in the GitHub issues or on Telegram and on Twitter.
Awesome. Thanks, Sean. And to your point, we're excited to be a part of a much larger open source ecosystem with many incredible tech partners, some of whom have even agreed to be sponsors for this hackathon. We very much appreciate them. And we've actually got 2 folks on the call right now, and the possibility for a third one to join in a little bit. So as a part of this closing presentation, one of the things that we would love to do is just quickly pass off the mic to, let's say, Jurij to start, to tell us a little bit more about Origin Trail and why decentralized knowledge graphs have the potential to be important in the scope of scientific research and the larger bioagent stack. Jurij, would you mind coming off mute and maybe just giving a few words on that?
Yeah, thanks, Eric, for the intro. So maybe just a quick kind of recap of what Origin Trail Decentralized Knowledge Graph is. It's an open source project, right? It's an open source foundation for building verifiable AI agents. And in the realm of DeSci, what this really means is that it enables builders, scientists, researchers, anyone who really wants to contribute to the DeSci space to transform any type of scientific research data, right? Papers, health data, any type of other. So we're here, actually, I'm here in Brussels right now, where we're showcasing another example where genomic data is used for a similar purpose. So to really transform that into a semantically rich and connected data on the decentralized knowledge graph that really provides the knowledge with 2 characteristics. One being that the knowledge can be connected across, or comes from and can be connected across different data sources, right? So we're not operating in a silo anymore. And at the same time, that knowledge graph that is being built from all of these different knowledge sources can always be verified in terms of source, right? So you can always find what piece of the knowledge was issued by whom. And that gives it a lot of power. And in this hackathon, builders were pretty much building agents, providing them with new ways to interact with the decentralized knowledge graph. So add data to it, add knowledge to it, retrieve knowledge from it. And why this is, I believe, an important component of the DeSci stack is because as this knowledge grows, right, and it's continuously connected, the more autonomous and the more powerful the agents can become. Ultimately, also themselves starting to add research to it, right, test hypothesis and so on and so forth. But before this happens, and for this to happen, you do need a solid, trusted foundation, and that's really what Origin Trail is all about. Yeah, I'm really happy to see the cool agents that the teams built based on Origin Trail, and I'll stop here.
Awesome. And we were also lucky enough to have Ziga, another one of the co-founders of Origin Trail, actually live at the BioXAI or the DeSci Berlin event. Got the pleasure of hosting a panel with him. Really smart guy. So we are lucky and grateful to have you guys as partners. Thanks for coming on. We're excited to continue building with you. We have another one of our tech partners and sponsors here, Nisita from ELIZA Labs. Nisita, is there any way you could come off mute and just tell us a little bit about ELIZA itself and why it's an important part of the bioagent stack?
Yeah, thank you, Eric. Super excited to be here. I'm Nisita, part of the ELIZA Labs, which is the team that actually supports ELIZA OS. The website is elizaos.ai. You can also find us on GitHub if you look up ELIZA OS. We're an open source community, like Eric mentioned. And ELIZA Labs, we're really trying to make sure we're providing the support to builders and folks to contribute better. We're continually looking to add, and this is something Sean mentioned as well, adding tooling and functionality, because that's what really makes ELIZA be useful with the bio community and pretty much any use case you can really think of. So I would think of tooling and functionality in the context of plugins, or that's what we call them within our ecosystem. So as a user or as a builder, if you come across an opportunity, we'd love to hear more about what are those additional tooling and or functionality, because we do believe, and I think that's where this hackathon's been super helpful, because the bio community, broadly speaking, was able to help identify a lot of these opportunities. Because I do believe this, in a personal capacity as well as professional capacity, that folks that know their craft are going to be the folks that help us identify what these opportunities are. Like for folks that do research in the biospace, you'll be able to identify opportunities in a way that, maybe I might not be able to. I think so. This was a fantastic example of that. So I'd say, please continue leveraging ELIZA OS framework. And if you're, listening to this and or watching this and maybe haven't had a chance to tinker with this yet, I would highly encourage looking it up, because it does allow you to do a couple of different things. Because we'd love to see builders and we want to help people participate and contribute in a way that truly benefits themselves within the bio community, as well as just the overall community, because there's always going to be opportunity for cross-pollination of things that might work in a different industry that might be beneficial, and vice versa. So thank you so much.
Absolutely. And thank you so much for being involved in the event, for being an active partner, and for helping participants at the hackathon and kind of taking some of those opportunities to further integrate ELIZA and bio. We really appreciate the team. We're excited to be working with both of the folks who are on this call and with every other sponsor who was involved. I do want to give a particular shout out to Solana, who was the main sponsor for the event. Jerry has done an incredible job with coordination, with working with us, with trying to go back and forth with some of the hackers that are there. We even got the pleasure of hosting Solana Accelerate as the hackathon midpoint check-in. So getting to go to the Solana offices in New York and kind of see the larger DeSci space, and especially some of the teams that are building on Solana. I think Vincent, who's actually on the call right now, is the co-founder of SpineDAO, one of, in my opinion, the most exciting projects on Solana today. So love to be a part of all of this ecosystem. Thank you to everyone else who was a sponsor here. And without further ado, Sean, I think it's about time that we start talking about winners. If I go ahead and share my screen, For those of you who may be following us on Twitter, want to go ahead and post this bioprotocol status with the different teams who we think have done an incredible job. But let me go ahead and share my screen. And Sean, if you wouldn't mind just quickly talking through some of the winners that we see down below.
Yeah, perfect. So let's start it off with these guys here. So I'll just go in order here. We have 4 main track winners. These were like some of the best hacks of the event. So Side Effect Net is a way to, it's a dashboard for, they used a custom data set pulled from the FDA on side effects of drugs and how they interact. And they've set up a very nice dashboard for if you or a loved one is taking multiple drugs, maybe for different illnesses, it's a way of seeing what side effects interact and what possible alternatives there are, as well as bringing in AI to have an extra opinion on more details of those interactions. So that was really cool to see. With Quantum Parse, it was a really cool application of like computational chemistry, where it's able to run inside ELIZA OS as a new action, and it reads these very specific chemistry notations and lets you ask follow-up questions in plain English. And so both for undergrads learning chemistry, for people in the lab, it helps the computational modeling and solving chemistry equations that really just help people work faster or teach them things. And so that was an excellent project. Microbiome DAO submitted something called the Biome AI, which is a way to interact with a Discord agent that you basically, there's a common test that you can take that tests your gut biome, and you get back a 50-page PDF with a lot of data. And these don't give a great recommendations or much more information beyond a very scientific listing of certain things. And so you can upload this to Discord, and the AI is able to talk you through it. You can ask questions about it, and it's able to help you out and basically give you life advice and for fitness, for dietary habits. And that was an excellent project. And maybe my favorite plugin is, it's called Holy Bio MCP. It's actually a set of plugins, set of 5, that turn, there's basically 5 specialized MCP servers where it's around genomics, longevity, drug discovery, and then interactions between drugs. And it's just these really powerful plugins where the ELIZA ecosystem is very nice, because once you have a plugin, you can just switch it on with a single line of code, and then you're able to, the agent is able to understand, because it has access to these actions, it can read, like internally the agent can read what they are, what they do, and then call them. And in this case, it gives them very powerful abilities for working with biochemistry tasks and biomedical tasks. So really impressed with these 4 projects. And this is, you can find them in the Twitter thread here. We had a Solana track. These 3 projects each win $5,000 for a really some really interesting hacks related to the Solana ecosystem. I could be using Sol directly and using programs in the Solana ecosystem. There are programs that they custom wrote. And so Tanya is about finding hidden null results between like in a knowledge graph or in a set of papers. It's detecting whether there are some phrases or some data points that the authors kind of gloss over and ignore, because one of the problems in science is that people don't want to really publish null results. They're not incentivized to. And so this is a nice tool to if you are a researcher, though, it saves you a lot of time if you know that someone else had a null result for a specific experiment that you're about to do. So it's almost like a public good to publish null results, but not many people are doing that. And so this is a really cool solutions for that. The Deep Idea project it's a way to like go through the idea maze for a new project or if you think you have a therapy or a startup, a business idea, this helps you navigate that. And they had custom contracts on Solana as well. And lastly, really impressed with the hypothesis to experiment orchestrator. They call it the HEO, that also built custom programs to accelerate this basically engineering discoveries using CRISPR. So it's an agent that is able to reason about like gene editing and protein engineering, and it reads papers, reads literature papers, PDFs and text, and is able to come up with hypotheses. And from those hypotheses, it comes up with a series of steps for an experiment. We like doing an experimental assay and testing it. And it's super cool. It's really pushing the boundaries of what ELIZA can do and what the bioagents can do. And so we're really impressed with this project as well. And yeah, thanks to all these teams. And we have a few more. As Eric said, we had a midpoint prize because we had a very long hackathon. Some teams really started out of the gate really fast and did some incredible work. So the biohack CLI from ValleyDAO is both a command line interface as well as a front end that is able to help you. So it's actually very similar to the Deep Idea, but any person can use AI assistance and research assistance to talk about like the total addressable market, the business plan, like all these things that a scientist will want to use in trying to get funding, but may not know the actual specifics, or take a long time to research the specifics without talking to an agent. So the ValleyDAO project is very helpful. We're going to be using it inside the DAOs, inside of the bio DAOs, as both telling people that they should be going through this process to kind of flesh out the different parts of their idea and make sure it's working well. And then we have the Protein Bank MCP, which is, for anyone not familiar, MCP is a kind of a new communication paradigm, kind of like an API for an agent to talk to some backend server and be able to do certain things and take certain actions on that server. And so in this case, there is a protein bank that is able to, a very large protein bank that an agent would want to access to understand what's already in there and read from it, and either use that in its experiment planning or tell the user chatting with it about and answer any questions they have. So this was a really cool integration and almost necessary. Like, these types of plugins are almost no-brainers for new scientific projects to add to their existing agents. And they're just, with ELIZA, you just turn them on, and then the agent is able to do these powerful new actions. And so wrapping up, we have the Kronos project, which was a team. There's these ancient Indian texts, I believe like Tamil language texts from thousands of years ago, that they went to a local library in India and scanned the text, added it to a knowledge graph. And these texts were around like the vertebrates and spine. And so in bio, we have a new DAO called Spine DAO, where they think like the older knowledge is still valid. And so these agents can go through a huge trove of ancient knowledge and see what's in there. And we really think that's a cool idea, and it has been found to be useful. So really impressed with everyone's projects here. And we had, I think, there's one special project here. The two of these are, I think, extras, but the neural nexus is a way to, let's see there. It's like connecting the molecules to like neurological drug discovery. And so they have a way to screen compounds and analyze them in 3D. And so for this specific discipline of neurological drug discovery, this tool seemed very useful, and used ELIZA as ELIZA plugins as well as a dashboard. And so I was really impressed with this team, and congrats, everyone.
Absolutely. And I did see one quick typo up here in the midpoint track. So just want to clarify, Chronos was 10K and the Validal CLI was 5K. But overall, we, as I mentioned at the beginning of the call, have been blown away by the number and the quality of submissions that people put forth for this event. We are beyond grateful for everyone's work here, and are excited not only to reward it, but hopefully to continue collaborations looking into the future. I'll tell you, my favorite part of this entire hackathon was the Twitter stream where we were hosting the midpoint event, and the two guys from Chronos, the SpineDAO project, were presenting live, detailing how they had gone library to library across rural India to collect some of these scrolls, which had never been digitized before. As we were live on that call, researchers from Yale reached out to these two kids and said, "Hey, here's my email. Let's get in contact as soon as possible." And if the idea that two people straight out of college can go through, add this kind of impact to the world, helping to digitize ancient manuscripts, and immediately be rewarded by straight contact with Ivy League researchers, that is still, to me, one of the most beautiful outcomes of this, and something that really embodies the ideals of decentralized science as a whole. So I would love to echo Sean's statement and just give a huge shout out to all of the different teams who participated in this event. Each and every one of you, even if you didn't necessarily end up winning a prize, we still appreciate the work and hope to see you as a part of the community moving forward. This is, I think, the first hackathon, but especially with other teams that are hosting their own. Bio is getting more and more involved in the space. So do want to give a quick shout out to Protocol Labs, who we are actually sponsoring a hackathon track and prize with in their event that has actually already started. Believe the signups close on June 7th, or on July 7th, excuse me. But one of our judges from the event, Slava, is now official representative of Harvard Dataverse, and has put a track online all about trying to bring decentralized identifiers and other kind of deep Web3 technologies to a new Harvard Dataverse instance. So very excited to see what happens there, to see how we can use these larger hackathon events to continue pushing the mission of decentralized science, both from a very human and from a very technical standpoint. So if you were involved, we would appreciate it if you would stay involved, and would love to see you as a part of the wider community. So with that being said, for the last about 20 minutes of this call, we're actually going to go through two, if we have time, Vincent will talk about Cronus as well, two different hackathon projects which we found exceptional. The first one being Mealy from Microbiome, and then we're going to talk about the guy in the VR headset, Stanley from Rare Compute, and hopefully get demos of both. So Mealy, if you wouldn't mind, can you go ahead and get us kicked off? Maybe share your screen, present a little bit of what you're doing in Microbiome.
Yes. One moment. Share screen. I'm Melia. I'm calling in from Berlin, actually. I just heard my Airbnb that I'm calling for is right next door to Molecule's bio office. I didn't know, randomly. But now that I know, I want to visit. I'll try to get there tomorrow. But
We would
can you
love
see my
that.
screen?
Absolutely. Not quite yet, but it may be coming up. I think it might be something
The
with the
browser
screen.
can't
behind the
access.
scenes.
Share screen.
I'm still just watching Stanley turn in 7 different directions all at once on his VR headset. It's quality entertainment.
Yeah. What about this? Yes.
There we go. Thank you. Much appreciated.
This so we built the Biome AI, which is essentially an agent to help you understand your microbiome report. And we built it as a Discord bot. I'll show you here. It's Biome AI V2. Hello. The reason we built it as a Discord bot is because you can add it into the DAO Discord, and you can see how different users are interacting with their microbiome reports, and it generates a lot of discussion. I'll upload now this sample report for BioAI. This is 3 megabytes of like highly technical stuff. I myself am not, I've been more the dev on this project. I've worked a lot with the microbiome team to understand the science of it. And now the agent can help me understand the science of it. So based on this massive, gigantic report, it creates a thread and creates a knowledge base on my report. So a RAG basically in the background. And it will combine this technical data that I got from my microbiome report with my now inputted lifestyle data. Like you'll see how it starts asking questions about my diet and different relevant lifestyle topics. And then when combining these two with some magic of prompting, you can get some good insight, honestly, to what type of hypothesis, what type of solutions would be good for your personal situation, what would be helpful. And then you can test that, take another microbiome test, come back, test again, and iterate for your own health. So it asks, did I take any antibiotics? No. This is very important because if you take antibiotics, it messes up your whole test. And also it asks when I took the test, but let's see if it get it. It then tries to predict what my diet is based on this report. And I could say, does this match? Yes, but I also eat a lot of ice cream. And this is all to get the most important information that is then used to get whatever suggestions to me. Like if I have any allergies, it's very important to know that stuff before suggesting anything to a user. I have stable energy throughout the day. And the fun thing about doing this in Discord is, as a thread, anyone else in the server can come in and look at this thread and then start also joining the discussion. But what about that thing, and going deeper? So it's a multi-user experience, makes it more fun and more interaction. So are these my digestive symptoms? Yes, that's actually it. This is all based on the report. And if I could just describe here whatever symptoms I am experiencing, and it would then add it to the full context. So then it takes this context of my lifestyle data and it has the RAG. So now I can read through this, like I get these insights, what I probably should do in my life: increase dietary fiber, moderate ice cream consumption, and well-diverse diet. And from here on out, it's a Q&A. I can ask any questions like, are you sure I can't eat any ice cream at all? Or whatever. I could ask a very technical question about this 50-page report because it's all in the RAG and it will find the relevant context about my specific situation based on my data. And anyone else can join in on this conversation if they wanted to in the server. So it's a fun thing to share. This is basically it. I could go on here doing Q&A, but let me see if you have any questions. Thanks for listening.
Absolutely. Yeah, my first question would be the thing about ice cream. Are you sure that that's not a bug? Because if that's the report I'm going to get back, I'm not going to use it. Sorry.
I'm not sure what it did answer. You don't have to cut out ice cream entirely. Just moderate your intake. So it was.
I still don't like it, but I'll take it. We do have one question in the chat. This comes from Slava, who's one of our judges, asking, how do you know if it's not hallucinating in answers? Can you track back the right triples from the knowledge graph from the answers that were put out?
This is excellent question. And to be fair, we still need a lot of more testing and finding these hallucinations. We have, I think there's one check, you double check that is this answer factually correct, but you can do more of that, definitely. And a lot of the juice in how this works is in the prompt, which was, I honestly got a lot of help from the microbiome team. What kind of focus the answers in a way that it keeps it safe and not venturing far out on, for example, suggesting diet changes that are against your allergies. But that's definitely future development, to put more of these hallucination checks. It's a good point.
Absolutely. And I know that that's something Slava is very passionate about, especially with a lot of his work in knowledge graphs. We even have our own knowledge graph expert, Jurij, on the call. So if you need any help working to implement some of those features moving forward, we've got plenty of resources to support you and any other developer on the call who is interested in working towards more hallucination-less AI moving forward. We did have one other person just quickly join the call. I see Akash, you're on the screen right now, and just wanted to give you a very quick chance before we jump over to Stanley's presentation to introduce yourself, maybe say a little bit about your background and what you hope to achieve through Bio@AI.
Yeah, thanks, Eric. Hey everyone, my name is Akash. I'm one of the co-leads of the AI team at Bio, along with Sean. And I'm just super excited to see the range of projects that we've received as submissions for the hackathon. Millie, I will probably be reaching out to you to see if I can use the Discord bot that you mentioned, because I'm probably there's probably a lot of product market fit with me on this app. I'll just leave it that way.
Not a fan of ice cream, huh? No,
I'm
really
a
cool.
I'm a fan of ice cream. I just don't know if my body is.
I understand that. Thank you so much for sharing, Millie. It's really cool to see, and I think a lot of people can benefit from this technology, and hope to see you around the community and hopefully helping to kind of teach others with some of the work that you've done, because it's great stuff. So with that being said, we are actually going to move to our next presenter. This is the man in the VR headset, the myth, the legend, Mr. Science Stanley. Stan, you want to take it away? We're having some problems with your audio.
Now?
There we go. We got you.
Yeah. All right. Always something. So yeah, the audio's okay, or good to go. All right. Forgive me if I keep the headset on and don't greet you guys properly. Definitely got it ready for demo, but that's always something you don't want to mess with. So anyway, so excited to tell you guys about our project. Let me just share my screen and we'll dive right in. So anyway, going to start with the scientist that this project was intended to support, a very brilliant molecular engineer named Amelie Schreiber, who I think is a pretty accomplished molecular engineer and very notable because she does most of her work in the open source I always joke that there's at least 10 startups right now fully funded using Amelie's code, and she's just given the stuff away. And there's a particular topic in biological machine learning that Amelie is sort of the tippy top of the field on, and that would be what we call in silico directed evolution of protein sequences. We're all familiar with GPT, these transformer-driven models that are really good at learning sequences. But to a computer, the sequence hits the CPU as ones and zeros. It don't know if it's French or Spanish or amino acids. So these same architectures that are having incredible impact in other areas can actually learn to speak proteins. So cool. And it isn't just like lip service to say they learn to speak protein, because on a mathematical level, proteins do have a semantic organization. Proteins have what you might call parts of speech. There's parts of a protein that interact with other parts. You might think of those as the noun. There's structural things like backbones that connect different pieces. Those might be your your adjective or your interstitial words. So it kind of turns out that proteins are one of the most learnable things. And so this Hugging Face post, which you can find, goes over a really interesting way to take a known protein, apply one of these models, and generate some new proteins. It's so fun. More than that, we actually have a lot of ability to control the evolution. So, for example, if we find a data set that is characteristic of evolution of proteins in a particular ecosystem, we can kind of put our finger on the scale and generate proteins that look like they evolved in that ecosystem. So, an ecosystem that has to deal with large temperature changes would potentially generate more thermostable proteins. If you had a data set of a particular category of, say, proteins that are present in an oncology, in a particular cancer, you'd be able to tune proteins that were more like those proteins and could interact and target those proteins better. So punchline, just like for linguistic agents, having the artifacts of agentic memory, the sort of tuning sets, the prompts, critically important to do good protein engineering. So that's what you're seeing right here on the screen. This table is a protein tuning set. So this scientist Amelie has found a large number of proteins that have a desired property. She's processed them into this table, and then this can be used to tune one of these evolutionary protein systems to produce proteins that are more like what we want. And yeah, just in the past year at Rare Compute, Amelie has discovered a novel gap junction opening peptide that will be entering clinical trials as a heart arrhythmia medication with Dr. Michael Levin at Tufts University. We've been able to raise the thermostability of a known plastic degrading enzyme 2 degrees, which is a pretty big deal. And yeah, we also will have kind of another really exciting announcement soon on our next therapeutic, which is just about to pass animal models. But all of this stuff, though, does take a lot of work. Each line of that table is an entry of a system called the Protein Data Bank. I love the Protein Data Bank. You hear people from DeepMind and AlphaFold talking about how exciting their work is, but like all of us, they stand on the shoulders of giants. This PDB Databank archive contains the aggregate work of the scientific community, imaging these little protein structures over the course of 40 years. And so, I think very important to remember that although we might be doing DSci, there have been people doing incredible open and collaborative science projects for, a long time. But yeah, how do we access it, and how do we organize those data sets? And that gets us right here to the protein MCP, Protein Data Bank MCP. We'll be putting this up on GitHub and inviting others to mess around with it. But yeah, it lets us access things like this cool protein here. The way it functions at a high level is we dump papers in one side of the system. And agentic process kicks off where each paragraph is read by a reader, reviewed by an editor. The paragraphs containing descriptions of proteins are sort of selected. Those are then put through a process that finds the protein, describes it, and then prepares a query for the MCP. So the MCP can take each identified protein, grab the structure file, grab all the supporting metadata, and then get it all, chopped up into one of these data sets. It's a pretty cool system. And my goodness, does it really save a lot of time? Because here, for example, you can see the project, which will be our first application of this tool. And that is the total paper archive for the incredible project Quantum BioDAO. So these are all the papers that the team at Quantum BioDAO have selected as containing interesting, potentially quantum proteins. And yeah, if we wanted to go through this by hand and produce one of these core data artifacts for protein engineering, we would have to go through this by hand, read all the papers, make entries. And yeah, instead, like we do just kind of put it through this pipeline. So anyway, system running real well. And then tomorrow actually we'll be at the quantum biology laboratory building our first example of this kind of fair, compliant, agentic memory bio data set. And yeah, this is kind of what it ends up looking like. Produces this kind of structured metadata object that describes everything that a human or an AI scientist would need to know about this protein. And then, gets it all ready to feed to one of these tuning sets. So, these molecular design and tuning projects often are very iterative, and you sometimes realize you're not getting the proteins you need. And you have to go, I think of it as like mix some new paint out of some different data. And, this takes that process from a week to 10 minutes. So, I think doesn't just accelerate the work of these scientists and save them time, but it also sort of changes the nature of the work. It really allows the scientists to see the protein changing in real time, and then sort of be able to understand how the proteins that they're adding to the mix are shifting things around. And so at first might seem a little gimmicky, but I just have pulled up this really cool 3D protein visualization system that we've interfaced. And so basically, like, you could just even verbally tell the system, go find proteins that are more like this, go find proteins that are more like that, and it would prepare all of them for you to inspect just like this, which honestly is pretty fun because, like, for example, here we have a protein that is believed to be the protein that many animals use to detect magnetic fields. So, we're all familiar with sharks use magnetic fields for hunting, but it turns out actually a lot of animals use magnetic field sensing in some of their biology. And so here we have this kind of like loop of protein structure that contains these ligands, and then this becomes the magnetic sensing apparatus, sort of the differential of magnetic field intensity on the different ligands creates a bit of a force that can be detected by the structure of the protein, and then that signal can be, amplified into the biology. So yeah, it is just kind of from the perspective of looking at proteins as these really remarkable, nano robots. Seeing them physically is fun. But yeah, I would just kind of end by saying also very useful because working on proteins requires biological input, machine learning input, input from many different specialists. And having this like sort of shared and shareable source of 3D truth, critically important for understanding, but also to create the rich data annotations that drive these systems. We could have a scientist come in and say, oh, this is doing this, this is doing this, this is doing this. And then all of that could push to the ontology that we've, had generated for us by this PDB system. So anyway, thanks so much for letting me show this off. Has quite frankly been a little seat of our pants because we're building it quite fast. But tomorrow at the Quantum Bio Lab, I think we'll be shooting some content, demonstrating an end-to-end, conception, generation, and deployment of one of these artifacts. So anyway, guys, now we'll take the headset off so I can actually formally give you guys a morning wave and a smile. And thank you so much for having me here. And more than anything, thanks for this hackathon. This has been so cool to be a part of, but also to see the community activating. So Eric, I know you've been busting your butt, man, and I'm really grateful to you and your whole team for everything, bro.
Yeah, it's insanely cool, Stanley. Both the VR, like Iron Man like manipulation with your hands, but also the quantum magnetic field. I think one of the things we're working on at Bio is the realization that both some new papers and like scientific advancement is coming from more like single author papers where one scientist can be aided by AI to do things faster. But then on the other side, there's also more citizen science where we can have a progressive increase in knowledge through like Origin Trail knowledge graphs where multiple people can be contributing to the knowledge graph over time and adding hypotheses and testing them quickly, both with like DAO funding and finding ways to tokenize these agents so that they can be used by many people, but also have the funding to do, because like all the demos, like there's going to be so many more experiments, but also the compute cost. And so finding ways to almost like crowdsource and tokenize these agents is what we're all about.
Absolutely. And just want to say thank you so much, Stanley. That is such a cool project. I couldn't even tell it was by the seat of your pants. That demo went so smooth. So very, very cool stuff. We appreciate having you here. Thank you for the demo and congratulations on the hackathon prizes. And thank you also for the work that you've done kind of helping to build the decentralized science space over the past 5 years and the work that you continue to do day in and day out. It's always inspiring. Now, with that being said, we do have 1 minute left on the space. So I know Vincent has been up here for a little while. To anyone who needs to jump off, be it from one of the folks who's already on the stream, from anyone who joined on YouTube, thank you for the time. Thank you for the attention. Thank you for coming and supporting this incredible effort towards building fairer, more democratized, and robust open source science in a community setting. We hope to work with you again. We hope to see you again. And it's been a pleasure. And with that being said, our encore for this particular Twitter space, if he has time, or this particular live stream, Vincent, would you have a quick chance to talk about the Kronos project? You're on mute if you're speaking.
Well, just a minute to congratulate you, BioFam, for doing what you're doing, because it's the true revolution. I mean, you're building bridges between across generation, across continent. You're creating new bonds between people, and that's basically what we need in this world. And I think we are you are regenerating the world with these great ideas. So basically, the quest, the Indiana Jones quest and Kronos it's just the beginning. But we have teams now from Latin America, from Thailand, from India, from France, and it just began with one of the oldest society of surgery, the French Surgery Society. The birth date is around 1731, and you need to know that this society has a phantom wallet. So we are giving them a spine token to reach out their undigitized gems in their library. And yeah, it's a real honor to participate to this journey with you, BioFam, and to mix old gems, old knowledge to this bioagent. So yeah, the quest is not over. It's just the beginning. And thank you so much, and thank you for all the sponsors to support us. Thank you so much.
Absolutely. And that's a great point I hadn't even thought of. We've got Stanley with the VR headset, and we've got ancient scrolls from India all combined into the same event. This really is spanning centuries, millennia even, because I think Stanley might be a couple of years ahead of where the rest of us are right now. So absolutely fantastic. Thank you for joining. Thank you for saying, and congratulations on going from, I believe it was one team to start with, to now having 4 or 5 all working towards the same effort. It's amazing to see the growth. And with that being said, Sean, Akash, I'm pretty much good for this space. It's been a pleasure. Maybe one of y'all want to come on and just quickly close us out.
Yeah, I've just had my bio agent spiel, and I think the new agent frameworks are incredible. The scientific tools are increasingly incredible, and we're just combining them in a new way, a novel way. And there's a lot to do, and you can just basically anyone who's listening, any hackers, any developers, they like any parts of this over the call today. You've seen many different aspects, from knowledge graphs to plugins to specific computational tooling. And so there's a lot to help with. And we appreciate all the hackers that had some amazing projects. And it's been an amazing 2 months, and it just feels like we're just getting started. Thank you, Nasita and George, for joining the call. And I look forward to working with you guys more. But with that, I think we can end it here.
Sounds
Bye, everyone.
good. Everyone.
Thanks.